during his presentation titled Overcoming the Undruggable Nature of the Most Common Human Oncogene, KRAS. Please welcome Dr. Shokat.
Thank you, Matt and Lillian, for inviting me to this opening session. And it's a real honor to be part of AACR during these challenging times. So it's a pleasure to tell you about progress in the last decade or so targeting KRAS. And before I start, I have a few disclosures to let you know and tell you about KRAS. KRAS was the first human oncogene to be identified in the early 1980s, and since that time, we've known that mutations in K, H, and NRAS constitute about 30% of all cancer. But there were no approved therapies for targeting KRAS until 2021, and that's the sort of point of my talk today, to tell you how the progress was made in that direction. Now, you might think that almost 40 years for a drug to be discovered is maybe typical, because drug discovery is very challenging. But when we look at just the immediate downstream effectors of KRAS, like BRAF and PI3 kinase, BRAF mutations were discovered much later in 2002, and just 9 years later, vemurafenib was approved for targeting V600 mutants in BRAF. So when we identify mutations in really actionable targets like kinases, then the drug discovery process goes very, very quickly. You might also be noticing that in this diagram that BRAF and PI3 kinase bind ATP, and you know that competitive inhibitors of ATP are behind many of the 100 or so kinase inhibitors that are approved. And KRAS binds GTP, another nucleotide, so it should have been possible to use the same strategies against kinases to attack the GTP site of KRAS. But the problem identified very early on in the biochemistry of KRAS was that the GTP affinity is 20 picomolar compared to about 20 micromolar typical Km for kinases, both protein and lipid kinases. So we would really need a drug that binds to the target in the picomolar range, which is very, very rare and probably impossible considering the high concentration of GTP in the cell and competing with that drug. So the most obvious pocket on this small GTP was really off limits from the very beginning. Nonetheless, once the discovery of mutations in KRAS were discovered, people like Doug Lowey's lab identified strategies to prevent KRAS oncogenesis by preventing localization of KRAS up to the membrane. Once this approach was put down, people realized that if you could identify the enzyme that added the lipid, the farnesyl group, to KRAS and inhibit farnesyl transferase, you might have a great therapy for treating KRAS mutant tumors. One of the first unfortunate steps in the history of KRAS drug discovery, however, was that although farnesyl transferase was eminently druggable and multiple drugs from multiple companies were developed to inhibit farnesyl transferase, in the particular KRAS paralog and the splice variant 4B, this farnesyl transferase inhibition did not work because of a bypass mechanism of another enzyme, geranyl transferase, another lipid-modifying enzyme that could recognize the unfarnesylated lipid tail of KRAS, and this bypass mechanism led to complete resistance to farnesyl transferase of KRAS inhibitors. So this really put a real damper on the strategies, and at that point, there were really not great approaches for treating KRAS mutant tumors or even ideas to go for that. That led into the era of many, many powerful mouse models that were designed and executed by many, many hundreds of labs to try to identify a therapeutically actionable target downstream of KRAS that could lead to inhibition of KRAS mutant growth, but preserve a therapeutic index by sparing the rest of the signaling. One of the most, I think, insightful sort of mouse models in this regard was done by Mariano Barbacid's lab, where he had shown previously that MEK, knocking out MEK or ERK, while they would shrink tumors, also had very, very profound toxicities when knocked out of the whole animal. However, one particular model, when he knocked out CRAF in established tumor models, the tumors that were dependent upon KRAS and had KRAS mutants in the cell disappeared, but the other paralogs, B and ARAF, could cover in the rest of the body for that loss of function of CRAF. So that really provides a very, very attractive approach right now for going downstream of KRAS and potentially coming up with a therapeutic index. The problem right now is a chemistry one, and as far as I know, but hopefully at the meeting this week, we will see somebody that has come up with a strategy for degrading CRAF in a paralog-specific way. That is something that I think is a high-value target. So other approaches to target downstream were really investigating the importance of KRAS to PI3 kinase, and an early mouse model generated by Julian Downward's lab, where he introduced a point mutation at the interface on the PI3 kinase side that binds to KRAS. A mouse model that was created with that mutation did not support any KRAS mutant tumor growth. That really was exciting because there were other inputs to PI3 kinase that were still preserved. So this looked like a way to selectively disrupt KRAS mutant activation of PI3 kinase. And until just very recently, there had not been an approach to sort of take that genetic approach into a pharmacological strategy. But Frank McCormick's lab with the NCI-RAS initiative, Lawrence Livermore lab, and BridgeBio have come up with a molecule that does sit at that interface, binds to PI3 kinase, and prevents KRAS mutant activation. This one is just in the clinic, and there are other companies like Vividian that have also developed molecules in this area. So this is also another area to be to focus on. I think it's been a long road. We thought maybe traditional PI3 kinase inhibitors or traditional RAF inhibitors would work in RAS mutants, but for various reasons, those strategies haven't worked, but these much more sort of mouse model directed strategies are really, really exciting right now. So let me return to KRAS itself and tell you how KRAS functions. As a GTPase, it binds to GDP and GTP. In the GDP state, it's off. In the GTP state, it's on. And what we mean by that is in the GTP state, the shape of the protein, those two colored regions on GTP, have a shape that's complementary to PI3 kinase or RAF being recruited up to the membrane. And to go from GDP to GTP requires a guanine exchange factor to destabilize that very, very high affinity picomolar binding of nucleotide. Once the open state is present, GTP binds and initiates the pathways I've told you about, but also sets the timer. The protein has its own internal timer through intrinsic hydrolysis that slowly hydrolyzes GTP to GDP, but the cell has other even more exceptional control over that hydrolysis rate, but using the GTPase activating protein that comes up to the complex and greatly accelerates hydrolysis. And you've seen the glycine 12 mutation noted on the cartoons previously. Now I can tell you that exactly how that glycine 12 mutation causes unregulated, dysregulated GTP stability in the protein is that it sits right at the interface of the GAP protein. And this is pioneering structural biology from the Wittinghofer lab that identified the key residue on the GAP protein that is pushed away because of a G12 mutation. So now that you understand how the G12 mutation causes transformation, now we can start to see what opportunities there might be in drugging this protein. And because it's a steric block that the glycine 12 mutation causes, it's actually very fascinating across different cancer types what that residue turns into in the different cancer types. So these are the 12 most lethal cancers in the United States. Green is incidence, and yellow is mortality, and in blue is the fraction of patients, each of those cancer types that have a KRAS mutation. So you see in lung, it's about a third, in colon, it's about half, in pancreatic, it's greater than 90%. But interestingly, the glycine 12 mutation turns into aspartate, valine, or cysteine in pancreatic cancer, Similar sets of residues in colon with cysteine growing in a little bit and aspartate decreasing. But in lung cancer, cysteine becomes the dominant mutation because that's the smoking-induced lesion, a transversion mutation. And so when we looked at the whole spectrum of this across all patients that have KRAS mutations, we saw that aspartate dominates clearly. And it would be great to go for a drug against the most common variant of the most common oncogene. However, we noticed that the cysteine is present in a significant fraction, and cysteine offers a great chemical opportunity. Because of its nucleophilicity, we can use covalent chemistry on the drug to take even weak reversible binding and then turn it into a covalent bond, which is irreversible, and hopefully keep the drug on the protein. The other feature about cysteine is that it's rare in the proteome, so there are not as many cysteines, so if we get a chemical reaction, it's likely to be more selective than against very common residues. So this is the strategy we took to make a covalent bond against the G12C mutation. The other feature that was very attractive is that if we used covalency, we would exclusively bind to the cancer mutant in the tumor cell and not the wild-type KRAS. So the other strategies that I highlighted, CRAF selective degradation or blocking PI3 kinase binding to KRAS, those really require extraordinary therapeutic indexes because of the paralogs covering. In this case, we would just spare the wild type with the drug itself, and it would be only mutant specific. So we set out to do this. We solved the crystal structure of the G12C mutation in the GDP state. You see the two switch loops are open in this case, and there's no obvious pocket on the protein, just as many and all of the structures that had been solved previously showed us. So then we needed a starting point. This is often the most challenging part of a drug discovery campaign. How do you get that first molecule, however weak it is, that binds to your target? There I was very fortunate to work with my colleague Jim Wells at UCSF, who had pioneered a very, very bespoke kind of fragment-based drug discovery method called disulfide tethering. This is a chemical library that has all members of the library have a disulfide, and then they have a drug fragment, a very small sort of piece of a drug on them. They do exchange reactions onto the cysteine, and if the member of the library has a fragment that's complementary to a sub-pocket, very small pocket, no matter how shallow, next to the cysteine of interest, then this molecule will be stable relative to other disulfides that are formed. And then as a control, beta-mercaptoethanol is used at higher and higher concentration to try to test the stability of this disulfide. So we ran through this collection of just 500 molecules, and this by comparison to many drug screens, which are now billions of molecules in DNA-encoded libraries or trillions in the sort of computer-generated libraries, this was a very, very small library. And we found two molecules that bound to the G12C form of the protein at 100%. They didn't bind to the wild type, which had other cysteines. So we had the cysteine selectivity that we needed. And I won't go into this too much, but I'll mention that the early molecules did not bind to the GTP state of the protein, which, if you remember why the G12 mutation is transforming, it's because it locks the protein in the GTP state. So the fact that we bound to the GDP and not the GTP state was a big concern, and we spent many, many years sort of uncovering that challenge and actually uncovering and realizing why this was nucleotide state specific came when we found the first crystal structure of this molecule bound to the protein. And that was shown here, which really revealed the attachment of the disulfide to the G12C position, and the rest of the molecule was bound in a pocket that we didn't see in the protein in the APO without the drug. So this is a case where the drug actually helped form its own pocket, and it formed it under what we call the switch 2 loop. And so therefore, we really needed this drug to find this pocket. We couldn't do it on its own because of the flexibility. They also told us why this part of the protein, why this drug was selective for the GDP and not the GTP form, because in the GTP state, this switch 2 loop would be folded into the pocket where you see the drug now. That turned out to be a very, very important point for us. We optimized, and in these early stages, we were never able to achieve GTP binding. But as I'll show you later, the field has overcome this challenge, and new generations of molecules do have GTP binding. So we had a molecule that we converted into an electrophile that works on drugs, a so-called acrylamide. So we took the disulfide, put the acrylamide on, and we have a molecule that was quite flexible, and I tried to impress upon you that the target itself is very flexible. So two flexible things trying to find each other is a very huge entropic cost. So we really needed to leverage chemical design and structure-based design to improve the affinity of the molecule for the target. We did that with my colleagues at AACR Pharmaceuticals. They developed more and more high-affinity molecules based on structure-based design. They came out with this molecule, ARS-1620, which was, I think, very, very important in the series of molecules because it's the first one to show animal efficacy of shrinking tumors. And that really motivated many, many other groups to also pursue the target based on the scaffold. And groups at Amgen and Mirati came up with variants of the molecule that made it into the clinic. They all share the same electrophile. They all share the GDP-only binding. So these molecules sort of wait for the cycle of GTP to GDP to happen and then trap the GDP state and then starve the tumor of the GTP state. And luckily, these two agents showed efficacy in lung cancer patients with G12C mutations. There are more agents in the clinic. I'll just give you one that is in phase 3. This is from Genentech Roche, divarasib. And this is a somewhat typical clinical waterfall plot from these patients treated with G12C inhibitors. You see there's complete response, about 80% disease control. At the lowest doses, there were progressive disease, and you'll see that your eyes notice this patient who benefited at first, but then had progressive disease. And this is the sort of state of the field currently. We have a lot of new KRAS drugs I'll tell you about more in a moment, but some patients become resistant, and how do we deal with that resistance? Either upfront combinations, better drugs, or strategies like you'll hear from Paul Michel, the next speaker, about overcoming resistance to targeted therapy. So now, about 10 years ago, we realized that cysteine mutation, that part of the pie, was quite well in hand, and a number of agents are in clinical trials, some that are more GTP binding. But the field wanted to go for the rest of the pie, and I think one strategy that was taken early on by Mirati Therapeutics was to essentially take off the electrophile, get rid of the covalent chemistry, and make the molecule that bound in the pocket that I showed you higher and higher affinity, but reversible affinity. And so if you had a molecule like that, you would have a pan-KRAS or a pan-RAS inhibitor, and you could go after the rest of the targets. We realized companies were pursuing that, and we decided we needed to take some approach that was sort of less common but might have an ultimate benefit that if we could overcome the chemical challenge would be desirable. And that was to stay on the nucleophile approach. But the other nucleophiles that were formed in the somatic mutants, serine, arginine, and aspartate, are much weaker nucleophiles, orders of magnitude less than the cysteine, and they're much more common. So we have this problem of how do we get a molecule to bind to a very common residue that is not very reactive and do so selectively. We overcame that after about 10 years of trying, and we used small ring systems that have ring strain in them, and that's the potential energy we used to go after the weaker nucleophiles. So we did serine, we did arginine in a more unusual way where we had 2 electrophiles, so we hit 2 of the nitrogens on the arginine side chain. But I think the most important one is this aspartate reaction that again used the small ring system But in this case, there is a chemoselectivity difference where serine attacks the carbonyl and aspartate attacks what's called the beta carbon. This is the carbonyl, that's the alpha carbon, that's the beta carbon. They both do the ring opening, but through different sites of attack. So I'll jump ahead and skip about 12 years of very, very frustrating chemistry and no reactions to give you the first molecule that gave us 100% reaction on the most common variant, G12D. And it showed you it attacks this carbon, it opens up, it makes an unusual bond to a protein, esters. We don't usually see drugs bound to proteins using an ester linkage. But what was amazing to us was that it reacted 100% with the aspartate at the 12 position, but if we moved the aspartate to the 13 position, it didn't react. By similarity, if we made another control molecule, protein, where we took the 12 position and made a glutamate, which doesn't occur in cancer, but it's a great chemical control, it also didn't react. And the wild type has 26 other glutamates and aspartates, it didn't react. So when we finally got the reaction to work, we realized that these weak nucleophiles, like aspartate, require incredibly high precision orientation of strain systems. Once you get that right, you get the desired reaction, but not the other reactions. We solved the crystal structure of that bound. We see the ester bond between the aspartate and the drug and the context. And these molecules, as I said, really, this part of the molecule came a lot of inspiration from Mirati 1133, which those of you who work on KRAS, you know how foundational that molecule was. But attaching the electrophile is what we focused on. So then we ran into a problem, which is that the molecule was not very stable in PBS, and it actually disappeared very quickly, making a sort of a big roadblock for us. But we, Chin-Heng and Ziyang in the lab, who were the two postdocs in this project, realized that there was a special feature of this ring system we were exploiting, that water, which was giving rise to attack and the decreased PBS stability, attacked the carbonyl position, whereas the aspartate had added to this carbon. And because nucleophiles adding to electrophiles have incredibly precise attack angles, we knew that the water attack angle was very different from the attack of the aspartate. And so we tried to play the game of sterically hindering what we didn't want while still allowing the reaction that we did want to happen. And so we added a methyl and an isopropyl group to the ring system, and this allowed us to prevent water attack and then still preserve aspartate attack. The isopropyl worked better than the methyl in terms of the stability, and the methyl and the isopropyl still allowed aspartate to react. So this was very, very exciting. And then we made a whole bunch of more improvements to really get the molecule tuned up for the cellular and animal efficacy studies. We made a number of other substitutions that I won't go through, again, inspired by Mirati, inspired by electrophile chemistry that we developed in our lab. And this led to molecules that were exclusively G12D selective cell inhibitors. They didn't inhibit G12V cells or other cells. So this was very nice, and we actually showed in this very early molecule animal efficacy of tumor growth inhibition. So we're continuing to improve this, changing, and now that we know aspartate is targetable, we're attaching new kinds of electrophiles that have other features that improve potency and efficacy. So to summarize, I've told you about starting off with the cysteine and how going after maybe not the most common, but the most chemically tractable mutant in a series really gave rise to, I think, a really exciting approach to target KRAS. I told you about leveraging G12D covalent chemistry. That has been a long journey, but something that has, I think, proven that is possible. The field as a whole in the last 10 years has gone incredibly fast and come up with incredibly innovative molecules to target KRAS, and I wanted to highlight a few of those. One of them is from Alessio Chiuli's lab at Dundee with Boehringer Ingelheim, and you'll see in this picture that they've made a molecule that bridges between KRAS G12V and the VHL E3 ligase to make a so-called PROTAC that can degrade KRAS. That had been a long goal of many people in the targeted protein degradation space, but because of the challenge of the druggability, that was always challenging. But now that molecule is in the clinic, and it's very exciting to see a way to actually just completely degrade KRAS as a therapeutic strategy. Another strategy that emerged from Revolution Medicines is another kind of glue molecule that actually brings together the top molecule is cyclophilin or immunophilin, and the drug itself was shown in green, and it brings cyclophilin to RAS in the GTP state. So its name has the designation on RASib rather than just RASib. So these suffixes tell you whether the molecule is exclusively GTP binding or GDP. And if you do the surface model, you see the whole protein is covered. But in cutaway, you see the drug sits in between the interface of the immunophilin and KRAS and makes contacts, the protein as well as the drug. And very excitingly, Pirolito's lab at Sloan Kettering showed that this complex is actually a synthetic gap and stimulates hydrolysis. So if you remember the problem that the G12 mutation causes, this molecule actually partially, completely reverses that biochemical defect. Very, very exciting. So lastly, just tell you who did all the work. John Ostrom and Ulf Peters were graduate student and postdoc in the lab who pioneered the G12C work. Jim